Good morning. I'd like to start off by thanking Dr. Stephen Emanuele and the Filio Group for having me speak at the pre-conference symposium today and to share this presentation with you. The purpose of the presentation today is to give you a broad overview of the ruminant immune system and to discuss some various factors associated with the immune system and how that can be influenced. As depicted in this slide, the immune system is generally broken down into three broad categories, which are natural immunity, innate immunity, and adaptive immunity. Natural immunity primarily refers to the physical barriers of the body, such as skin, hair, mucus, tears, our stomach acid, and digestive enzymes. The innate immune system primarily refers to the inflammatory response and associated immunological non-specific cellular activity associated with phagocytosis and natural killer cell activity. While depicted as separate entities in this slide, natural immunity and innate immunity are often grouped together when we discuss the first line of defense of the body. Adaptive immunity, on the other hand, refers to specific cellular activity associated with T cells, B cells, and antigen-presenting cells. While each of these components of the immune system have their own specific role to play with regard to providing immunological protection, it's important that all three components of the immune system must be fully developed and functioning properly to provide adequate immunological protection for the animal. It's important to realize that natural immunity is not a constant entity, and in fact, it can actually be influenced by numerous factors, including wounds, dehydration, nutritional deficiencies, the genetics of the animal, and even stress that the animal is exposed to. The innate immune system, which we consider to be the inherent portion of the immune system that the calf is born with, is actually the first response to any pathogen invasion to prevent infection. Innate immunity also represents what we refer to as the antigen non-specific defense mechanisms of the immune system, which is listed by the calf after exposure to an antigen. The innate immune response and the adaptive immune response each have unique characteristics regarding their ability to recognize and react to pathogens. For example, the innate immune response is a very rapid response that occurs within minutes to hours after a pathogen invades the body. Because of its ability to recognize thousands of highly conserved pathogen-associated molecular patterns, the immunological protection provided by the innate immune system is very broad. The primary function of the innate immune system is to actually control the spread of infection until the adaptive immune system is activated, and to direct the development of the adaptive immune system. As this slide depicts, the innate immune response is controlled by very specialized cells such as basophils and mast cells, neutrophils, eosinophils, monocytes and macrophages, dendritic cells, and natural killer cells. This slide depicts the rapid time course associated with various biological markers that are released during an innate immune response elicited by exposing cattle to an endotoxin challenge. As depicted in this graph, the stress hormone cortisol and the pro-inflammatory cytokine TNF-alpha increase significantly within the circulation within minutes following exposure to the endotoxin. Likewise, the calf generates a febrile response within minutes following endotoxin exposure. The innate immune response, while highly conserved in mammals, is not a constant entity, and it can be influenced by numerous factors, including physical and environmental stress, social stress, nutritional stress, psychological stress, as well as immunological stress. Unlike the innate immune response, the adaptive immune response is slower in onset. Biological markers associated with adaptive immune system activation are often not detected for days or even weeks following exposure to a pathogen. However, the immunological response by the adaptive immune system is a specific, customized response to a particular antigen. The primary function of the adaptive immune system is to identify invading pathogens and clear them from the body through various B cell and T cell activities. Another very important aspect of the adaptive immune system is to direct the development of the immunological memory response, thereby causing a more rapid and stronger adaptive immune response to subsequent exposure to the same pathogen. This slide represents the classical memory response, in which the primary exposure to a pathogen stimulates the production of low-affinity antibodies to a specific antigen, and subsequent exposure to the same antigen stimulates a more rapid and high-affinity antibody response. Unlike the innate immune system that generates a rather nonspecific immune response to a wide array of pathogens, the adaptive immune system is capable of mounting an immune response that is specific to a given pathogen. The adaptive immune system is also very diverse, mounting an immune response to bacteria, viruses, fungi, and parasites. Another distinguishing aspect about the adaptive immune response is its ability to tolerate self or host cells and to recognize and destroy non-self or pathogenic cells. Adaptive immunity can be divided into either passive or active immunity. Passive immunity, or otherwise known as maternal immunity, stems from the transfer of antibodies from the dam to the offspring through colostrum during the first 20 to 24 hours of life. Some antibodies that the calf receives from its dam will be directly absorbed through the intestinal wall, while others will stay in the gut to attack pathogens locally before they migrate into the calf. While we typically consider colostrum intake during the first 24 hours to be crucial to the calf, the time frame is actually much shorter, as 75% of the calf's ability to absorb antibodies is lost by 4 hours of age. This table, taken from a study by Selk, highlights the importance of timing associated with colostrum intake and circulating antibody concentrations. For example, if feeding is delayed by only 6 hours after a calf is born, the antibody absorption capacity is already reduced to 66%. Delaying feeding for another 30 hours after birth results in approximately a 90% decline in the calf's ability to absorb antibodies. It's important to note that the immunological protection provided by passive immunity is limited and is highly dependent upon the concentration of antibodies absorbed through the colostrum intake. As these graphs depict, the amount of colostrum intake determines the subsequent circulating antibodies in the calf, and the lower the concentrations of antibodies earlier in life can significantly influence the overall survival of the calf. After passive immunity wanes at about 7 to 8 weeks of age in the calf, it's critical that the active component of the adaptive immune system kicks in to mobilize the necessary cells and produce the antibodies required to provide subsequent immune protection for the calf against invading pathogens. Even with both passive and active immunity, the calf is still vulnerable to pathogen infections and disease. One of the highest risk periods for the young calf is the period between when passive immunity is waning and the active immune system has not fully developed. This is a period of time when various management practices can be crucial to the health and overall survival of the young calf. Diving a little deeper, we can classify active immunity as either cell-mediated or as antibody-mediated. Additionally, we can further categorize it as either natural or artificial. Natural refers to exposure to pathogens within the calf's environment, whether that's from non-animate objects or from exposure to other calves. And a good example of artificial would be antigen exposure through vaccination. As its name implies, cell-mediated immunity refers to the immunological response that is mediated by immune cells that act directly against pathogen-infected cells within the body. Cell-mediated active immunity requires the involvement of specialized black blood cells, called T cells and B cells, along with chemical messengers known as cytokines and antibodies that provide long-term immunological protection. T cells, which originate in the thymus, provide what is referred to as cell-mediated immunity and are classified as either T helper or T cytotoxic cells. As their name implies, T helper cells help T and B cells grow and divide to produce more cells to fight future infections. Cytotoxic T cells, on the other hand, are capable of acting directly on the pathogen-infected cells in the body and destroying them. B cells, which originate in bone marrow, work in concert with T helper cells to help produce large quantities of antibodies, which are specific to a particular pathogen and provide long-term immunological protection. Humoral immunity, often thought of as antibody-mediated immunity, involves macromolecules such as antibodies, complement proteins, and antimicrobial peptides that defend against extracellular pathogens. Antibodies play several roles in providing immunological protection, including binding and inactivating pathogens and their toxins, activating the complement system to destroy bacterial cells, and facilitating phagocytosis of foreign substances within the body. To summarize, adaptive immunity can be either passive or active, either naturally or artificially induced, and either cell-mediated or antibody-mediated. Unique to the adaptive immune response is the ability to generate a customized response directed towards a specific pathogen. However, generating this specific immune response requires time. Therefore, the adaptive immune system is slower in onset as compared to an innate immune system. Therefore, having a fully developed and properly functioning innate immune response is crucial in order to allow the necessary time for the adaptive immune response to be effective. As mentioned earlier in this presentation, the immune system is not a constant entity, and it can be influenced by numerous factors such as natural variations between animals, like gender, animal temperament, and genetics. As many producers are aware, animal health can also be influenced by exposure to various environmental stressors, such as cold stress and heat stress, as well as dietary manipulations. However, less commonly perceived factors, such as exposure to prenatal stress or pathogens in utero, can also have significant and lasting effects on the immunity and overall health of the cattle. Over the years, our group has been investigating various factors that influence the immune system and overall health in cattle. As we have continued these efforts, we have identified all of the factors listed on this slide as potential mediators of immune function. Likewise, we have often found that there are interactive effects among many of these factors. For example, we have documented that the innate immune response is significantly different between common temperamental cattle. However, there are also significant metabolic differences between these cattle. Therefore, while the differences observed in the innate immune response may be genetically driven, it may also be significantly influenced by the metabolic differences between these cattle. We have also documented that there is inherent sexual dimorphic innate immune responses between bulls and heifers, as well as heifers and steers. We have documented these differences in several different biological markers, such as the rectal temperature responses depicted in this graph following an acute endotoxin challenge. We have also documented sexual dimorphic responses following a bovine respiratory disease challenge model. Collectively, our studies have demonstrated that the onset of the immune response following an immunological challenge is delayed and sometimes blunted in heifers as compared to bulls or steers. With the increased scrutiny on the use of synthetic antibiotics in livestock production, our group has been extensively involved over the years in evaluating various nutritional supplements that may prove to be beneficial with regard to boosting the immune system and overall animal health. While we've demonstrated that some nutritional supplements such as zinc, bacterial pre and probiotics, and yeast-based products can have beneficial effects on the immune system, we're continually striving to understand more about how nutritional supplements influence immune function in cattle. Several years ago, the observed metabolic differences between common and temperamental cattle led us to ask the question, can we modulate the pro-inflammatory response in cattle by altering circulating concentrations of lipids? To address this question, we took 21 Holstein steers and divided them into 3 groups of 7 calves per group. The control group, represented by the green line in this graph, was administered saline at 0.5 milliliters per kilogram body weight immediately prior to the LPS challenge. The dextrose group, represented by the blue line, was administered a 50% dextrose solution at 0.5 milliliters per kilogram body weight immediately prior to the LPS challenge. And the third group, the lipid group, represented by the red line, was administered a 20% interlipid solution at 0.5 milliliters per kilogram body weight per hour from 1 hour before until 6 hours after the LPS challenge. These results clearly demonstrate that not only additional energy, but the source of energy the calves received can significantly influence the fever response to an immune challenge. In addition to the different fever responses noted in the calves, we also observed significant differences in several biomarkers associated with the stress and the innate immune responses. For example, this graph depicts the differential circulating cortisol responses that we observed. Interestingly, while the lipid-infused calves displayed a greater fever response, the graph shows that the cortisol response was actually blunted in those calves. Likewise, serum concentrations of tumor necrosis factor alpha, or TNF-alpha, one of the primary pro-inflammatory cytokines associated with the innate immune response, were also lower in the lipid-infused calves following the endotoxin challenge. As with TNF-alpha, serum concentrations of interleukin-6 were also significantly lower in the lipid-infused calves as compared to the dextrose-infused and the control calves. Given that TNF-alpha and interleukin-6 are considered the primary mediators of the fever response, and the fact that lipid-infused calves displayed greater fever responses to the endotoxin challenge, it appears that the source of energy being provided to the calves during a period of immune activation may be involved in the modulation of the fever response itself. Interestingly, we also observed significantly reduced circulating concentrations of interferon gamma in the lipid-infused calves. Given that interferon gamma is considered to be a chemical messenger that links the innate immune response to the adaptive immune response and helps to direct the progression of the adaptive immune response, it appears that the source of dietary energy can impact both innate as well as adaptive immunity. While further research is needed to fully elucidate the relationship between dietary energy and the source of that energy with overall immune function, these data provide insight into how dietary formulations could impact animal health. Another area of research that has been the focus of our group pertains to the impact of maternal stress on the developing calf. While we are all aware that stress and immune challenges can influence the growth, reproduction, health, and the overall well-being of cattle, we rarely think about the long-term consequences of these events on the developing fetus. For example, while we know that stress and immune challenges can alter nutritional requirements for the gestating cow, do we understand the consequences of these events on the developing fetus and the subsequent growth, productivity, and health of the calf after it's born? To determine if an immune challenge to gestating cows would alter the postnatal immune response of their calf, we took 50 crossbred pregnant cows and administered 25 of them saline subcutaneously, and administered the other 25 a subcutaneous dose of LPS at a rate of 0.1 micrograms per kilogram of body weight during the third trimester. When the calves from these cows were weaned at 205 days of age, we administered 12 heifers from each treatment group an IV LPS challenge to evaluate their innate immune response. The blue line represents those heifers whose dams received the saline, and the red line represents those heifers whose dams received the LPS. As this graph clearly depicts, the fever response of the heifers whose dams were administered LPS during the third trimester had a significantly reduced peak fever response, but a prolonged fever response compared to the control heifers. In addition to the different fever response, we also observed differences in TNF-alpha. The blended TNF-alpha responses depicted in this graph for the heifers whose dams received the endotoxin during the third trimester may explain the reduced peak fever response in those heifers. As mentioned earlier, both TNF-alpha and interleukin-6 are considered to be the primary mediators of the fever response. Likewise, the prolonged elevation of interleukin-6 depicted in this graph for those same heifers may explain the prolonged fever response observed previously. Therefore, while both TNF-alpha and interleukin-6 may both be primary mediators of the fever response, they may have independent roles with regard to stimulating and maintaining the fever response. Collectively, these data highlight the fact that prenatal exposure to stressors or pathogens may alter postnatal immune function and health of the calf. While we are rapidly discovering and learning more about the immune system and how various factors influence the overall health of cattle, we must still realize that there are other factors that we may not be considering or even thinking about that can impact the overall health of cattle. Additionally, we need to rethink how we view animal health. For example, in human medicine, we recognize individual variations such as age, gender, nutritional status, genetics, or even personality type can have a significant influence on the overall health of a person. However, too often in livestock production, we tend to address health more at the herd or group level, looking at generic factors such as pre- and postpartum status, stage of lactation, or where there are first calf heifers or multi-pairs cows. Perhaps we need to start viewing cattle health more from an individual basis rather than a herd or group basis, taking into consideration individual variations that we would consider in human medicine. Finally, we also need to consider that the immune system does not function in a vacuum. Overall animal health requires adequate energy and nutrients to fuel proper immune function, and the modern dairy cow is always at the edge of being deficient of energy and nutrients. Additionally, any additional stress or metabolic disorders can rapidly deplete these necessary resources. We need to remember that stress and activation of the immune system requires energy, and significant amounts of it, to fuel the necessary biological functions to return an animal to a homeostatic balance. Creating and maintaining a fever response alone is very energy intensive and can require a substantial increase in metabolic energy. And should an animal become septic, the energy requirements could be as great as 60% or more. This slide represents the typical blood circulating glucose response to an endotoxin challenge. As the graph depicts, when the immune system is activated, glucose is mobilized to provide the necessary fuel for the immune system, and this increase in glucose mobilization can be detected in circulating glucose concentrations. During the initial stage of immune activation, hepatic glucose output and the peripheral insulin resistance is greater than the immune system's utilization of glucose. However, With continued activation of the immune system, glucose is rapidly utilized at a pace that is greater than hepatic output of glucose, and the animal enters into a period of hypoglycemia. In fact, our research has demonstrated time and time again that when cattle are exposed to an endotoxin challenge, there is a rapid increase in circulating concentrations of both cortisol and glucose that occurs within minutes. While we often think of cortisol as an indicator of stress, we must remember the primary biological action of cortisol is that of a glucocorticoid hormone that mediates the uptake and utilization of glucose as well. When an animal becomes hypoglycemic, the amount of energy available to fuel the immune system becomes limited. In summary, we have to realize that in order to maintain a healthy cow, all aspects of the immune system must be fully developed and properly functioning. Each aspect of the immune system plays a critical role in maintaining an individual in a homeostatic condition, and any factors that jeopardize or alter any aspect of the immune system will have an impact on the overall health and well-being of that animal. Finally, when we discuss animal health, we have to realize that animal health can be compromised by stress. And while all stress, or the physiological responses to stress, are not the same, they do all require energy and nutrients. Utilization of energy and nutrients to cope with the stressor may limit these resources at a time when they are necessary for mounting an adequate immunological response. Additionally, we need to rethink how we view animal health. All cattle are not the same. Whether they look alike or not, there are individuals that have different requirements in order to survive and be productive. Thank you for the opportunity to share this presentation with all of you today. Should you have any questions regarding the information I've presented, or would like to discuss any of the topics or research data included in the presentation, please feel free to contact me at the email provided at the bottom of this slide.